Hello, everyone, and welcome to today's webinar, Fast and Automated TSA-based Multiplexed Immunofluorescent on LabSat Research. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Lunaphore. To learn more, visit them at lunaphore.com. Now, we encourage you to participate today by submitting as many questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Alexandre Kehren, an application development team leader at Lunaphore Technologies. Alexandre, welcome. You may now begin your presentation.
Thank you and welcome everyone. So today I'm going to present you the general overview of the microfluidic technology developed at Lunaphore and the autostainer LabSat. We're going to go through a specific application of LabSat, which is the automated TSA-based multiplexing, and see how to optimize the assay on tonsil tissue. And then we're going to see a method for easy transfer of those optimized protocols to cancerous tissue, and in this specific case, a study on non-small cell carcinoma. At the end, we will have a live Q&A, and as mentioned before, we will answer the questions received during the presentation. So, a Lunaphore core technology is based on a microfluidic chip called MTP for microfluidic tissue processor. It is used for tissue analysis assays like immunofluorescence or immunohistochemistry. The chip is equipped with a series of microfluidic channels used for reagent delivery. It is clamped against a standard glass slide that makes a closed chamber over the tissue sample. This technology is called fast fluidic exchange for FFX. So the pressure-driven reagent flows directly, almost instantaneously, in the chamber. The liquid exchange is very rapid between the steps. A heating element placed just below the slide allows precise temperature control at any time, as well as rapid temperature changes. The perfect sealing between the chip and the slide offers a controlled environment to the tissue with multiple benefits. Staining times are much faster due to reduced diffusion times in the chamber. There is a more uniform staining over the tissue. The rapid and high temperature epitope retrieval are made possible while preventing reagent evaporation and preserving the tissue. This technology was born at the Swiss Federal Institute of Technology in Lausanne and had been widely validated across a number of studies and publications. We have demonstrated that running microfluidic-based immunofluorescence assays with short incubation times, we could better quantify the fluorescent signal from the tissue thanks to the avoidance of saturation. We could even classify ambiguous HER2 status of breast carcinomas into FISH positive and negative, basing ourselves on the quantification of our fluorescent HER2 staining. We have also shown that we can obtain extremely rapid chromogenic and fluorescent stainings on different markers on both FFP and frozen samples. Additionally, we've shown that DNA and RNA can be preserved during the stainings performed with our instrument, and the samples can be further utilized after staining for NGS. With our technology, we can also run FISH assays and targeted RNA expression. And finally, in this presentation, I'd like to introduce you to the use of our technology and our instrument LabSat to perform tailor-made signal amplification-based multiplexing assays in combination with the Opal kit from École. The MTP chip is integrated in an automated device that is called LabSat Research. Reagents are simply loaded in 8 standard Eppendorf, and buffers are stored in 4 Falcon tubes. The chip is placed in a staining chamber maintained by a lid, ready to be clamped against a slide. Another important microfluidic element of the system is the distribution chip. It acts as a gateway between the reservoirs and the chamber and ensures the proper control of the flow towards the tissue. Reagents are pushed from the reservoir towards the distribution system, from where their dispense is controlled, to the staining chamber. Once the reagent is finished, reagents are simply discarded to a liquid waste. After having tested and proven the efficiency of the MTP on single markers immunofluorescence and immunohistochemistry, we decided to move towards multiplexing assays. There is an important drive towards multiplexing techniques, as the context of personalized medicine requires even higher number of markers to be investigated in order to build better diagnostics. Multiplexing immunofluorescence and immunohistochemistry in particular are interesting, as they allow for simultaneous marker detection while keeping the information of morphological context. That is especially relevant in the study of the interaction between the tumor and its microenvironment. So the method chosen to do immunofluorescence multiplexing on LabSat is the pyramid signal amplification, TSA. This technique allows simultaneous marker detection on the same tissue. The working principle is the following. So on your first antigen of interest, you would detect using a primary antibody that you would bind with a secondary antibody conjugated to an enzyme, here horseradish peroxidase, that in turn will catalyze the reaction of pyramid fluorophore conjugate that binds to the tyrosine residues at the antigen site. Before moving to the next marker detection, the antibodies need to be removed by application of heat on the tissue. This step is called elution. The covalent bond between the tyrosine and the tyramide ensures that the signal stays on the tissue, even after the elution step. Then, subsequent markers can be detected using the same method by applying a different fluorophore. This can be repeated for all the markers in a panel, and once all the tyramide are bound, the slide is ready for imaging. The TSA kit chosen for this project is the widely used Opal kit from École Biosciences, and the goal was to adapt these reagents to the LabSat system. We used a 7-color manual detection for 6 markers plus conscious staining with fluorophores of excitation wavelengths ranging from 520 to 690 nanometers. So our objective is to automatize a full 6-plex using the Opal kit, and while the pre-processing, meaning deparaffinization, rehydration of the tissue, as well as imaging, will still be done outside LabSat, the machine will take on all the steps that are directly related to the staining itself, starting with the heat-induced epitope retrieval, protein blocking, primary antibody, secondary antibody, and the TSA itself, then elution, all the steps being repeated in a number of times corresponding to the detected markers, and finally contrast staining. The assay development process was organized around 4 main axes. The first one, the single-plex optimization for each marker, with the objective to reach good and repeatable performance for every marker individually. The second axis is the uniformity optimization, with the aim to obtain homogeneous signal levels over the tissue, even over large sections. And then the antibody elution optimization to make the heat-induced strip-off reach complete antibody removal between cycles while preserving the tissue morphology at the same time. And finally, the multiplexing part, bringing all the individual optimized elements in one multiplexing assay and build a fast and efficient workflow on the machine. The 6 markers selected for the panel are typical immunomarkers, FOXP3, PD-L1, PD-1, CD68, CD8, plus pan-CK for cytokeratins. The optimization of the assay was done on FFPE sections of human tonsils, a positive control tissue for those markers. So first axis of the tonsil optimization project, single-plex optimization. So the assay was directly developed on LabSat with a goal to ensure high staining quality while keeping a reduced execution time. The parameters could be directly adjusted from the software interface of LabSat Research, and the parameters that were available for us to play with were the antigen retrieval, so both temperature and pressure settings, the reagent incubation, here again temperature, duration, and reagent flow optimization, and finally primary antibody and substrate titration, just like in any regular assay. The image acquisition was done on a Mantra imaging station, and the Inform software was used for image analysis. When using the OPAL detection system, acquisitions need to be post-processed. In particular, the signal needs to be deconvoluted to separate the information corresponding to each OPAL fluorophore. We built a spectral library prior to this study and used Inform to do unmixing, as well as cell segmentation of the tissue for further analysis and quantification. The single-plex staining optimization resulted in bright and specific signal for all the markers with high normalized mean intensity of balanced value across the 6 markers. The results are here displayed in software reconstructed bright field view, which is the Inform pathologic view. For each marker, the variability of the protocol was assessed on 9 consecutive slides. The single-plex repeatability showed less than 15% of the signal variability, underlining the effective automation of protocols on that system. Now the uniformity axis. Uniformity of the fluorescent signal is an important feature of an optimized protocol that can be challenging to obtain sometimes, especially on large tissue sections. LabSat offers some interesting functionalities that help obtaining a better uniformity. When using a standard incubation of the reagent on the tissue, depending on the method that is applied, this can lead to an inhomogeneity in the signal level across your tissue. LabSat allows for an active control of the flow during the incubation. The liquid motion in the chamber can be controlled at any time, ensuring a more uniform distribution of the reagent over the tissue. This method, called dynamic incubation, increased significantly the level of uniformity of stainings done on LabSat. We assessed the efficiency of dynamic incubation by looking at the signal gradient on slides incubated in a standard fashion versus the slides that were applying the dynamic incubation method. And we saw that over 1 centimeter of tissue, the signal drop was reduced down to 6% when using dynamic incubation, compared to 30% with a standard incubation, which represents a significant improvement in uniformity. The third axis of the project is the elution optimization. The role of the elution step is to ensure a proper removal of the primary secondary antibody complex between 2 marker detections. The method that we use on LabSat is simply applying a high heat to the tissue. To assess the efficiency of this elution method, we compared a reference stained slide versus an eluted slide, which simply consists in applying the elution process after primary antibody and secondary antibody incubations before re-incubating with secondary antibody and the substrate. If the elution is efficient, no signal should be observed on the eluted slide. The elution efficiency was defined as the signal removal rate. This elution efficiency was tested for all the markers in the panel, and signal removal rate was over 99% for all the 6 of them, which was considered as a performant elution. Since our elution process involves high temperature, it was important to test that the repetition of those heat cycles would have limited impact on tissue morphology. So we tested different repetition numbers of those heating cycles for 3 different single-plex, and we could observe no apparent degradation of the resulting staining due to the repetition of the elution. So nuclear morphology, extracellular metrics, and epitope detections were preserved. The last part of this tonsil optimization, it's the full 6-plex assay, and our goal is to build an efficient workflow with a minimum number of user interactions. So the tissue preprocessing and reagent preparation is handled by the user, that then has to load all the elements on the machine. LabSat then perform fully automatically 3 marker detections. Then, due to limited number of reservoirs, a user intervention is required, made protocol, in order to load the reagents needed for the last 3 cycles. After what, the protocol can be resumed and finished. Finally, the slide can be retrieved at the end of the protocol, mounted and imaged. The final result is a semi-automated 6-plex plus counterstaining that can be achieved in 4 hours and 12 minutes. The full assay showed adequate marker detection compared to the single-plex optimization. The conclusion of this first part of the study is that the LabSat offers a TSA-based multiplexing approach with high signal intensity for each marker, an adequate signal uniformity across the tissue section, an efficient antibody elution with over 99% of signal removal for the 6 markers, while preserving the tissue morphology and the epitopes at the same time. All this with a high level of consistency. So we're now moving to the second part of the study, which is the transfer of the multiplexing assay using LabSat on a different tissue type, a cancer tissue. So multiplexing assay optimized on tonsil can sometimes not be completely adapted to other specific tumor sections because of different marker expression patterns, or sometimes tissue specificities like different structures, different levels of autofluorescence, etc. The question we ask here is this tonsil-optimized protocol applicable to my cancer tissue samples? We approach this with a 3-step transfer strategy. First, we applied the tonsil-optimized protocol directly to our cancer sample. Then we evaluated the performance and defined what needed to be re-optimized. And finally, we modified protocol parameters in order to reach the desired performance. We're going to go through a small case study on non-small cell lung carcinoma, whole-tumor, and TMA sections. Our objective is to reach an acceptable performance on this tissue in a minimum number of optimization steps, because our aim is to avoid the re-optimization from 0. LabSat offers the flexibility of optimization, and some important parameters can be readjusted directly from the software interface, like the antigen treatable temperature, the incubation times, but also protocols are modular. You can add extra steps like blockings, quenching, etc. And of course, you can always play with the standard element of an assay like reagent titrations. So the first phase of the transfer strategy, we applied the Tonsil-optimized protocol directly on non-small cell lung carcinoma samples. IHC stainings of the corresponding tissues are shown here as references for marker expression. And we could observe that the direct application of the protocol on this particular tissue showed lower intensity levels than expected for all the markers compared to what we had on tonsil, which indicates that reoptimization might be needed. So we applied consecutively two simple protocol adjustments. The first one was to increase the antigen retrieval temperature of the assay, and the second one was to increase Opal 540 concentration. Opal 540 is the termite that we associated with CD8. And this was enough to rescue the signal levels and to show appropriate detections for all the markers. We then applied this reoptimized 6-plex to NSCLC tissue microarray in order to assess the protocol on various expression patterns. And here the reoptimized protocol showed appropriate detection of all the markers on those TMA cores comparably to the standard IHC reference. Various patterns of expression could be detected similarly to the IHC reference using this reoptimized protocol. And the total time of the assay remained unchanged compared to the Tonsil-optimized protocol, staying at 4 hours and 12 minutes. So as a conclusion, LabSat offers the possibility to run a 6-plex 7-color assay in a timely manner, with high quality results, both in terms of signal, uniformity, strip-off efficiency, and morphology preservation. The Tonsil-optimized protocol could be transferred to lung cancer tissue for the same assay duration. And the protocol transfer process was a simple two-step adaptation. I am now happy to answer your questions.
Thank you so much for that presentation, Alexandre. We will now start the live Q&A portion of the webinar. And if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's take a look. We already have some great questions coming in from our audience members. Our first question: what is the maximum thickness of the samples that can be placed in the microfluidic chamber?
Well, so we are working typically with FFP tissue sections that are ranging between 2 micrometers and 5 micrometers thickness. And we have tested up to 10 micrometers without any issue. So usually that's what we recommend. We want to avoid going higher than 10 micrometers.
Thank you very much. And is the antigen retrieval automated and performed on the device?
Well, yes. So the all the steps of the staining, starting from antigen retrieval, are taken on by LabSat. So it all starts after slides deparaffinization, and everything is taken on by LabSat stainer, and everything happens within the microfluidic chamber.
Thank you so much, Alexandre. Are the primary antibody used in commercially, is that available?
Yes, so LabSat is an open platform, and it's basically compatible with every commercially available reagent or kit. So for this study in particular, we have used off-the-shelf primary antibodies. And for multiplexing TSA assays, we have been used commercially available monoclonal antibodies.
And have you tested this with other non-Opal TSA reagents?
Yes, we did. We tested the Thermo SuperBoost TSA kit, which gave good results. And what was interesting is that the protocol that we had developed for the Opal kit was quite easily adaptable for this other detection kit.
Thank you, Alexandre. And I want to thank our live audience. These are such great questions coming in. What other applications are you thinking of to develop on the LabSat instrument, Alexandre?
Well, first, I think we are thinking about increasing the potentialities of the multiplexing TSA application. So we would like to increase the number of markers that you could detect using TSA techniques on LabSat. So we are currently working on a proof of concept to go up to 8 markers at a time on the same slide, while at the same time offering more options in flexibility and template adaptation, like more incubation times or different temperatures. And at the same time, in parallel, I think we are also working on completely different applications. So we would like to adapt the platform to the sequential immunofluorescence techniques. So sequences of regular primary, secondary fluo-labeled antibodies with imaging protocols in between each staining, which is a technique that can enable to reach much higher plex than multiplexing TSA. So this is currently under investigation.
Thank you so much. Now, could you scan the stained slides on the other digital scanners, and are there any specific requirements for this?
Well, yes. So LabSat performs all the staining steps of the protocol, and everything that's related to imaging has to be done outside of the LabSat. So I would say that it's compatible with any microscope that is equipped with the right multispectral camera. So for instance, here for this Opal assay, we used the École's Mantra and the image analysis by Inform for signal unmixing. But it has to be compatible with the detection kit and the method that is applied on LabSat.
Thank you so much. Now, how would you best use the LabSat, given that it's a single slide stainer?
So LabSat Research is not a high throughput instrument. It's an instrument for low throughput needs, for precious samples, or for single-shot antibody optimization. You could also use it as a workflow support or as a complement to other high-throughput instruments. For instance, you could use it to test new samples in parallel of while other stainers are busy. You could also use it for urgent stainings because speed of the assay remains the main asset. But in general, I would say that LabSat Research is more suited for project where it's not necessary to run 30 slides at a time, rather to obtain results fast, and also if you're looking for high staining homogeneity on samples where it is difficult to achieve.
Thank you so much, Alexandre, and thank you again for our audience. We have such great questions, and we have time for some more. How does the time of multiplex staining compare to larger multi-slide stainers?
So for typical 6-plex assays that can be found in the literature, so very broadly used, like Biomechanics, usually has a turnaround time in the range of 12-13 hours, which would be the same for the DACO Autostainer Plus, I would say 12 to 13 hours. So here in this study, we could run a single slide in 4 hours and 30 minutes. So yeah, if you're looking just in turnaround times, yeah, I would say it's 3 hours and 30 minutes against 12 hours.
Thank you so much. Now, could TSA be used for cochlealization analysis?
Yeah, so in this type of assays, or at least with what we could see on LabSat Research, it's an interesting technique for that. First, because you can detect multiple markers at a time. Also, because you can achieve subcellular precision. But for colocalization analysis, you have to make sure that the assay is correctly optimized, especially regarding crosstalk. So you have to make sure that spectral unmixing is properly done, that you do not get bleed through from other markers or the channels. So a good way for that is beforehand of the optimization, you could make sure to allocate markers that are supposed to colocalize to spectrally distinct fluorophores. That could be a way to go for colocalization analysis.
Thank you so much. Now, is the image analysis automatedly with the Inform software?
So for so we use the Inform software for image analysis, and this software features many different applications for image analysis. So ranging from spectral unmixing to split the raw image into different channels, to cell segmentation and cellular population classification. So the software has a whole image analysis pipeline that you can use to interpret your results.
Thank you so much. We have time for a couple more questions. Alexandre, how do you optimize the ratio of 6 or multi-markers after each marker is validated?
So there's transition to make from single marker optimization to the multiplex optimization. So basically what you want to make sure is that you do not perform differently when you're in the multi-marker staining compared to the single marker staining. So it is very important to make sure that your SBR hasn't changed, that you do not have bleed through from other markers in your single marker channels. And so TSA is known for such challenges, so it's very important to make sure that you do not mask the detection of your markers with the detection of previously stained markers. So of course there is a comparison of signal to background ratio to make between the single marker stainings and the multi-marker stainings.
Thank you so much. Now, have you tried staining to an CTC circulating tumor cells?
So staining in CDCs or in general on cell samples, cell pellets, is not something that we have tested yet, and it's not something that we consider is compatible with the current multiplexing TSA template on LabSat. It's of course something that we'll keep in mind for future development.
Thank you so much. And we have time for one more question. Do you have any ideas to increase even more the homogeneity of the staining? Could you imagine to increase the flow speed in one side of the chip?
Well, so it's so with the the dynamic incubation technique, it's we could already reach good homogeneity levels, but of course it's something we seek to continuously improve. So now we are looking to different ways of increasing homogeneity in the chamber rather than flow-related techniques. So it's still very early stage in the development, but something like temperature-induced diffusion or other techniques to enhance diffusion are currently being studied.
Thank you so much, Alexandre. And actually, we have time for a couple of more. Our audience is so great. We still have questions coming in. Is it good for the brain tissue? Is our next question.
So I would say you could follow. You could follow the same approach that we had for lung, in the sense that you would need to first see how does our standard protocol behave on your particular sample, and then establish in what way you could tweak the initial standard protocol to make it fit to your brain tissue. There, I would say that for brain, you might want to make sure that first your tissue thickness is compatible with the device, and then that you have a way to control the autofluorescence of your tissue. So for that, it's totally feasible to implement an autofluorescence quenching step and add it to the standard multiplexing TSA protocol. Is something that we have tested and that would be feasible here.
And Alexandre, what is the advantage then Leica's multi-staining bond RX?
Yeah, so of course, so I don't think you could directly compare those two devices, because Leica Bond RX is a 30 slides stainer. Here we are talking about LabSat Research, which is a single slide stainer. I would say that in general, for high throughput applications, so Leica Bond RX remains the standard. But if you're looking for a device that has very low turnaround time, if you have only a few precious samples to stain and you want to move fast in your project and optimization, then LabSat can offer you results in a very timely manner. And I think that's the main asset compared to Leica, but also to other available devices.
Thank you so much. Now, did you try to use a 9-plex update resend École system on the tissue sample?
Yes, so we tried that on LabSat. So it's still in development, so it's not commercially available yet as a template on LabSat Research. But the first test showed very promising results, and it works really well. So we were really satisfied with how reproducible, consistent it was compared to the initial 6-plex optimization that we ran with the old École kit.
Alexandre, thank you again for this great presentation today. Do you have any final comments for our audience before we go?
Well, I would say that as a take-home message, the main assets of LabSat Research regarding multiplexing TSA assays, it's the instrument versatility. It's a very open platform. You can use off-the-shelf reagents. You can adapt it to your own detection kit. It's also very easy to re-optimize the standard protocol to adapt it to your sample, to adapt it to your experimental plan. And once this is done, then you can take advantage of a LabSat main value, which is very short staining time and high quality results.
Alexandre, thank you again for your presentation and for your important research. And we'd also like to thank LabRoots and our sponsor, Lunaphore, for underwriting today's educational webcast. Before we go, I want to thank our audience for joining us today and for their interesting questions and questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, stay safe. Bye-bye.
Thank you very much.